1, 2, 2. Is it working? Test, test, test. All right. Hey, everyone. Welcome. So I want to welcome you all to the HS&T Hokie pitch. And just a reminder of what the really big prize is the impressive set of shark jaws, completely authentic. I believe, Zach, you caught this shark, right? Yeah. So there you have it. So this is actually our 7th year doing the HS&T Hokie pitch. And I have to say that the quality of the presentations and the competition improves and gets more intense every year. So we're really looking forward to the 5 teams we have competing. I just want to say some words of thanks to some important individuals. First up, I want to say thanks to our judges, Dr. Friedlander, Catherine Elam, the former president of the Junior Achievement Association, and did 20 years of service there, and really appreciate you having you here today, Catherine. Ford Kemper. Ford has been a long-term feature of this competition. He and his company, Woods Rogers. What are the other names I'm forgetting? Ford. Well, yeah. Yeah, they've generously supported this competition over the 7 or 8 years we've been running it, and as I say, Ford's a fixture of the competition, and just thanks for your support, Ford. And James Ramey, another long-term participant in the competition, as a judge, also as a lecturer. I think you've lectured in each of the 7 years that we've done it, and I know the students always look forward to your lecture. I say we've got a real life investor coming in, and then when it turns out that he's an awesome teacher as well, it's always a bit of a surprise for the students. Okay, so thanks, judges. So I've mentioned the support. I'd also like to thank Dr. Friedland of his support of the competition. Thank you, Mike. Very generous. Mike will also be supporting the reception that we'll be having after the competition. So I understand there's some alcohol out there, but I can't confirm it. It's in the middle of the day. Yeah. All right. So just a reminder. You have 7 minutes, and at the end of that 7 minutes, Zach will cut you off. So be ready for it if you go over. You'll have 3 minutes for questions, also pretty strictly regulated, although we may be a bit looser on the questions than the actual pitch itself. I also want to say thanks to the student IP mentors. Kenny Young, thank you, Kenny, and congratulations on your American Heart Association Pre-Doc Award. Yeah. Rahul Amin, Megan Sadovi, congratulations on your American Heart Association award. Mason Wheeler, where's Mason? Oh, there you are. Hey, Mason. You're always hard to miss. And Mary Fraser. Thank you, Mary. I'd also like to thank our business mentors. Unfortunately, a number of them couldn't be here today. Francis Farrell had to be called away. He's out of town. Cynthia, where are you? Cynthia Lawrence. Thank you, Cynthia. Where are we? Greg Feldman. Now, Greg is feeling unwell today. It seems like he has the flu, but he and I have been in contact, and he wishes his team and everyone else well today. Kevin Bloomfield is also not here. Kevin had to go to Blacksburg because his dad had a medical situation that he had to take care of, so he won't be here today as well. And then finally, last but not least, Mary Miller. And it's so good to see you again, Mary. I'd also like to thank our lecturers this year, including, I've mentioned James, but also Mark Van Dyke, who was a former faculty member here at Tech, but has since gone on to become a member of the leadership team at the University of Arizona. So he's a real grown-up now. Mark Bondry of Link Launch on main campus, and also Sarah Snyder, who was a former postdoc at the Institute, but is now the leader of Beam Diagnostics. So with that, I'm going to hand it over to Zach. The way this is going to work is Zach is going to be the person that regulates and chairs the competition itself. I'm going to slink into the background, or at least the foreground, and but I'll be timing. So, Zach.
Thank you, Dr. Gordy. So I'm pretty sure I just got this job because Dr. Gordy doesn't want to have to cut people off when they go over. So it's going to be really fun, I'm sure. So if everybody could just keep to 7 minutes and I don't have to do it at all, then that'd be great. So first, I just want to say thanks for getting to be involved in this. So I've been at the Institute for 4 years now, and I've been doing this competition in some capacity since then, whether my first year it was competing, being an IP mentor, or this year getting to actually participate and do a couple of lectures for the class. So yeah, last reminder, just keep it to 7 minutes. And our first team up today is Myocyte. So if you guys could come up and.
Test. Okay, good afternoon, everyone. My name is Emily, and I'm here with Ellie and Liang to talk about our company, Myocyte. So, personal story for me, hopefully don't cry. This is my cousin, Ryan. In this picture, he was a young 32, recently graduated from his PhD program, and he was hoping to pursue a career in education. He was recently married, and one night while he was at home with his wife, he had a heart attack. He was rushed to the hospital, he was assessed, and then he was later discharged. They told him he had something called NSTEMI and that he would have a few years to live. Unbeknownst to us and the hospital systems, he didn't. He passed away a few months later. Many people like Ryan go through our hospital systems with cardiac events, and they're told one thing, and then they pass away a few months later. So our company and our aim is to prevent this, to improve risk assessment for cardiac patients and working with clinicians and making sure that proper assessments are held. We are hoping to ask for $450,000 in your investment to help us implement our product and to work with hospitals to make sure lives are saved. So every year in the United States, 805,000 individuals have heart attacks. 70% of those are considered to be NSTEMI. That means that 563,500 individuals are at risk for complications. NSTEMI is a long-term chronic illness that many patients are told that they have in the hospitals. Of those patients, 9 to 20% of them go on to suffer cardiac death within 1 year, and there aren't a lot of great assessments that hospitals have in determining who those are.
So, which method do current researchers use to predict the cardiac death? Timmy and Grace are the two commonly used behavior indexes. Researchers often use linear regression models with Timmy and Grace as predictors to do the prediction. However, such linear regression model can only capture the linear relationship among variables. If there are more nonlinear complex relationships, such model fails and they have a very low prediction accuracy with decreased sensitivity and specificity. Moreover, such static behavior index is pretty static. They cannot capture any dynamic moment-to-moment fluctuations of body signs when patients are suffering from heart attack. Therefore, to overcome these strategies, our IP proposed a non-statistic base. We use the deep neural network based on a very dynamic, high time resolution electrocardiogram data to improve the prediction accuracy. As you can see from this figure, the deep neural network consists of multiple layers of neurons, which mimics our human brain. What we have to do is put our patients' relevant behavior and EKG indexes into such deep neural network, and it will automatically do the complex calculations and return the probability of the future cardiac death. The big advantage is that, unlike traditional CT models, which can only capture linear relationship, such complex neural network can basically account for any type of relationship, which greatly enhances the prediction accuracy. Moreover, we do not have to extract the features manually. The neural network will automatically extract relevant features by adjusting the weights of nodes in each layer, which can save us a lot of time and energy.
So to summarize, our technology is based on a deep neural network that interprets EKG variables in order to better risk stratify our patients. And this gives doctors and other providers the tools that they need in order to medically optimize those patients, whether it's with medications or for future interventions. This is a multi-billion-dollar industry. There are a lot of companies that are using artificial intelligence in cardiology. Most of these are based on real-time interpretation of EKG and Holter monitor data in order to better diagnose patients. Our company is unique in that we're actually using the similar data sets, but in order to risk stratify patients in order to assess for future risk of death. We're primarily focusing on urban community hospitals that care for a majority of cardiac patients around the country. To start, we will be looking at hospitals within the state of Virginia. In our pilot phase year 1, we're going to be applying for seed funding through the NIH, developing our web platform, applying for FDA approval, and then piloting our project with 2 hospitals in the state of Virginia. From there, we hope to scale to other hospitals in the region, and then by year 3, apply for Series A funding. This is a financial summary of our 3-year plan. Our financial model is primarily based on a subscription service. However, in year 1 with our pilot hospitals, we will not be charging a subscription fee, and therefore we will have a deficit. And that's why we're asking for additional seed funding now. Our 5-year projection shows that by year 2, we anticipate we'll be profitable. Our IP has around 13 years left on it, so we have a lot of longevity. We'll be able to scale to other hospitals over the next several years. Our team is uniquely positioned for this project, so we're all translational scientists. Liang has a specific background in machine learning, and I am an internal medicine physician. Many of us have been affected by the devastating effects of cardiac disease. We hope that with this technology, we'll be able to save lives and prevent future patients from slipping through the cracks. Thank you. Any questions?
Identified a very important issue and problem, no doubt. A gap and a need, great on that. And I think the plan on the back end looks reasonably quickly looking at it. So my question's more kind of in the middle about the details, and I'll try to just nail down 2 things. One, these kind of neural networks, of course, can be used for all kinds of things. And the magic is always in the secret sauce of that networking. So you're relying, as I understand it, primarily, if not exclusively, on ECG data. At one point, you had put the word behavior there in a couple places, and I wonder But my question really is, should you or have you considered the possibility of incorporating other types of data to strengthen it, such as, for example, echocardiography? I mean, there are lots of other things, but and if not, why have you limited it entirely to ECG?
That's a great question. And actually, this neural network specifically has been studied in several clinical trials where they looked at actual patient data. And in one of them, they used not only this neural network, but also BNP data and echocardiograph data. And they found that it improved the outcomes or improved the risk assessment for those patients. So adding in additional data can be helpful. And I do think that's something that we could incorporate into our web platform, certainly.
I want to add to that because I think the question is like, why do we only use EKG data, right? As I said in the presentation, one big advantage of EKG data is dynamic. It contains a temporal relationship. Like if the only behavior indexes like questionnaire, it describes the feeling, the body sign in a static period. That's an average sign. Can you get what I mean? But EKG is moment to moment. It's very fluctuations, highly dynamic. So we believe there is some important information during such high time resolution data. And the neural network can handle such temporal data information with temporal information.
Just to follow up on that, I didn't hear you say, will you get individual ECGs when the patient comes in, or will this be a Holter monitoring over time? You talked about the dynamics. And of course, seeing the ECG under active states at home in a natural ecosystem at rest, et cetera, provides a lot more information. So which of those two, which type of data will you collect?
So most of the time, patients with NSTEMI, it's a chronic illness is what it's characterized as. And so they're constantly going to the hospital for echocardiograms, physician meetings and things like that. And so it would be a long-term study on the patients when they go in for each of their visits.
So they would not be wearing Holter monitors. They would just, when they come in for individual visits, get an ECG. Is that correct?
I believe so.
The IP itself protects Holter monitor data as well as EKG. I believe it can be both. Most risk assessment models right now are when the patient hits the door. So we use Timmy and Grace model when patients come into the hospital, initial and STEMI diagnosis. This data is interesting because you could actually use it to trend a patient over time, even after they leave the hospital. So it gives us some longevity. And also it allows us to look back at prior data as well.
We can have one more question.
Can you clarify what the patent covers and what's to prevent those existing competitors from moving into this area?
Yeah, so the patent, it covers the machine learning for the dynamic evaluation of these ECGs data sets. It's looking at the computer's looking at the morphological changes of the ECG over time. So not just the normal waveforms of the ECG, but also in addition to that, how those waveforms change beat time to time. The patent itself includes the data set that they tested it in. So it was about 750 patients or so. There have been subsequent clinical trials on this data set after that point. The longevity of the patent, it expires in 2034. So we've got some time on it.
All right, thank you guys. All right, next up, we have metanostics.
Good afternoon, judges and audience. It is truly a privilege to provide you with this opportunity to you. My name is Elena Baltons. I'm Caitlin Stebbins.
And I'm Ozioma Chukwuchinaka. We are Metanostics. We believe knowledge is power. Today, we're here to ask for $3.5 million for 10% stake in our company.
We're asking for this investment for people like my grandmother. This is my grandmother here. She's truly one of the strongest women I know and has always been a mother figure in my life. Unfortunately, 3 months ago, we received the news that she was diagnosed with breast cancer. She gave me permission to share this MRI result, where it says that her lymph nodes showed suspicion for metastasis. My family and I have been reading these words for the past month over and over, yet we have no idea. There's no follow-up appointments yet, and we're not likely to know until early 2023. But our family is not unique. 2 in 5 Americans are diagnosed with cancer every year. Some of us in the room have most definitely dealt with this in some way, whether it be a friend or family member, or maybe even had to face a cancer diagnosis ourselves. Maybe some of us have even experienced a loss that can come with cancer. Cancer is the second leading cause of death in the United States, and 90% of these deaths can be attributed to metastatic cancer specifically. And it can take years after the initial diagnosis to catch metastatic cancer. My family is lucky that we can find out within a few months. But what if my grandmother could have found out the day she was diagnosed 3 months ago?
And what if it wasn't just Alina's grandmother, but every patient could find out in a single afternoon? Luckily, there's a solution. In 2014, researchers at Cornell University developed a microfluidic device to detect pancreatic circulating tumor cells. As you can see on the left, 4 patients were diagnosed with primary pancreatic cancer. And with this device, doctors were able to classify these patients as high risk for metastatic disease, which was then confirmed on surgical inspection before the patient showed any new symptoms. And this technology isn't new. It's been proven time and time again, but no device on the market offers high sensitivity. So what if this device could be brought to market through our company? And what if it could be used for nearly any cancer? With Metanostics, this can be a reality. Our microfluidic device offers the capability to prevent unnecessary screening procedures and significantly reduce metastatic cancer.
Now we understand that these are bold claims, but I'll explain our device to you. So it'll all start with a small blood sample that will be taken upon initial diagnosis or perhaps for a remission check-in. It would be a 10 milliliter blood sample. For reference, that's about this much. And then that blood sample will travel through a series of chips. The first detects separase, which is present in 90% of circulating tumor cells. The second chip is EpCAM, which is present in 65% or 85% of circulating tumor cells. The last chip would be an interchangeable chip for specific cancers with their corresponding biomarkers. Results would be shown on ELISA staining display, so physicians would know within the same day whether or not circulating tumor cells were present in the patient's blood. Positive tests would then be taken to a lab facility for further analysis utilizing the imaging window, so that way there's more clarity on the identity of circulating tumor cells. While having high sensitivity, our device is also highly affordable. It costs about $15 to make one Metanostics device, and with a selling price of $200, this allows Metanostics to have a wide profit margin of 93%, all while maintaining affordability for our consumers.
Now, the current competition doesn't hold a candle to Metanostics. We are the only company that offers a device with no radiation at a low cost that can be performed point of care in any office and requires no additional equipment for primary results. The only other device on the market right now is Cell Search, but they've only been validated in breast, prostate, and colorectal cancer, and at $600 per test are 3 times as expensive as Metanostics.
Our product aids in the decision of proper care by providing a quick and affordable way to detect cancer metastasis through Metanostics device. We plan to target for our market hospitals, cancer centers, and local clinics. For our consumers, our target consumers will be local clinicians, hospitals, and oncologists. For marketing, we plan to attend oncology conferences such as ASCO, creating partnerships with local distributors, and also going to outreach events locally and regionally. To deliver our product, we plan on creating channel partnerships with distributors such as McKesson and Henry Schein for the distribution of our product to our target market. We also plan on creating partnerships with diagnostic labs such as Quest Diagnostics for the analysis of positive tests. Our strategy is for metanostic testing to be the standard of care when dealing with cancer patients. These are our key milestones. Over the first 5 years of our product being on the market, we plan money will be raised through fundraising, attending pitch competitions, and also applying to grants such as the Pancreatic Cancer Translational Research Grant and the Early Detection, Diagnosis, and Prognosis Grant. During our 4th and 5th year, we plan on taking our product to the national and global market, allowing us to meet the unmet needs that many countries have with lack of access to PET scans and MRIs for the traditional diagnostics of cancer metastasis. Today, we're here to ask for $3.5 million for 10% stake in our company. 50% of this will be used for manufacturing and distribution, which is about $1.7 million. 45% will be used for R&D, which is about $1.5 million. leaving 4% for basic costs, which is a little over $100,000, leaving 1% for applications, which is $14,000. Here we have our break-even analysis. Our company estimates that we will break even by year 5.
Our metanostics teams consist of a highly comprehensive group of individuals varying across the translational spectrum, with specialties such as biomedical science, public health implementation, as well as business management. All our team is really missing is you.
So we are here today to offer you, our early investors, a debt investment of $3.5 million for a 10% stake in our company.
We know with your investment, we can help people like my grandmother and the millions of other Americans that are diagnosed with cancer each year.
Here are some words from clinicians. We want to thank you for your time today and your consideration. Thank you.
Have to go into a larger device.
Ideally,
Is
this
that
would be the
device in the
device.
market?
This would be the device that we would have in the market. Using the ELISA antibodies, we would have the specific markers there for separase that would be for 90% of circulating tumor cells. Currently, cell search only uses EpCAM, which is only effective 86%. So this would be the device in the market.
All in, meaning there's no syringe pumps or.
There would be a syringe pump included, and they included that in our cost and the next slide when they we included that with
Okay.
syringes as well.
So very nice presentation, very professional. I can't help but say this, there was a company called Theranostics, which I'm sure you've heard of. And so when you hear metanostics, and I heard you say this will detect any cancer, Theranostics jumped up in my mind, right? So the first question I have is on the third chip. Are you telling me that you have access to highly precise biomarker analytics for every cancer?
So an important differentiation is this is not a diagnostic test. This is risk stratification. So this is a tool for clinicians that allows them to classify patients as high risk or low risk. It's just another piece of information in the clinical decision making. So if you have a positive metanostics test, which can also include these FDA approved biomarkers that are currently in clinical use, a high-risk test would suggest more routine monitoring, possible surgical exploration, or additional clinical treatment. On the other hand, a negative test would suggest low risk for metastatic disease, and you could actually stretch out the routine screening that patients have, just hoping to prevent additional costs to the patients and to the hospitals.
And so taking that to the next step, potential interventions, for example, you find tumor cells X in this patient. What do you know about, number one, if that truly has metastasized, it's in the blood, but where it's going to take up residence? So many metastatic diseases from the primary tumor can end up in a variety of different tissues, and what would be the therapeutic intervention? So I guess what I'm really asking is, how actionable is that information, and how will you connect it to the caregivers to actually take the next steps?
So it really just depends on personalized medicine. So certain types of cancers are known to metastasize to specific sites. But in general, like I said, this is a screening tool. So if you have a patient who has a positive test, it's not necessarily going to tell you that they have metastatic disease or that it's going to settle in a specific location, but it can tell you that there's a higher risk of something else happening down the road. So this can increase the screening with PET scans, MRI scans, depending on the cancer, just so that you are able to keep a closer eye on your patients and then decide on a clinical intervention depending on what type of primary cancer you saw.
Just as additional, upon initial diagnosis, oftentimes the pancreatic cancer has already metastasized, but they've always diagnosed as just localized. If you took this blood test initially and you have indication that there's a high risk of metastasis, you can implement interventions much sooner than in traditional measures.
How far have you gone with this so far? Actually, 3 steps, chips. Who else are you working?
We have a list of where we plan to. I think get some materials in manufacturing. Where we would outsource the individual components, and then we would do the final assembly in our facilities, and then we would connect with our distributors. Thank you.
Metanostics, that was a really good presentation. Next up, we have AccuSight. You guys want to come up here?
Hello, we are AccuSight. Thank you so much for listening to our presentation today. Our company's mission is to provide quality, portable, non-invasive early detection and continuous monitoring of brain health to reduce the impact of secondary injuries and traumatic brain injuries, or TBIs. Our product is a ocular bioimpedance apparatus and electro goggles that will monitor dynamic changes in intracranial pressure and cerebrovascular autoregulation, which is critical to the blood flow and the supply of nutrients to the brain, and is often impacted in TBIs. 1.5 million individuals survive a TBI each year. That's 225,000 hospitalizations and 60,000 deaths. In the last year, 5.3 million Americans were living with a permanent TBI-related disability. In Virginia alone, the rate of disability was 19%. And the CDC estimates the total annual costs for this are well over $40 million. One of the problems with diagnosis is that CT scans can miss soft tissue injuries and can have rates of up to 58% missed TBIs. When I think about these rates, I think about Mel. Mel was a high school student who was ready to go off to college. He was in a motor vehicle accident and sustained a head injury. He went to the hospital, got his CT scan and other current measurements, and was discharged. After 72 hours, he returned to the emergency room presenting with neurologic problems. And that was because of a missed brain bleed, which led to long-term cognitive problems and emotional regulation problems. It also had a huge impact on his family financially. If he had had access to continuous monitoring during the time of his stay, they could have detected dynamic changes and potentially incorporated that into clinician decision-making and avoided this outcome. According to Grandview Research, the total global market for TBI monitoring devices was $2.9 billion. That's expected to grow annually by 7.5% to reach $5.5 billion by 2030. Of this global market, the US market makes up 53%. And of the US market, the 43% of it is made up by hospitals, ERs, diagnostic centers, and first responders. And that's why they are our target market.
The current gold standard to detect TBI is by ICP measuring using a catheter. It's done by drilling a hole into the brain of the patient and then inserting the catheter into the fluid-filled space, which then calculates the intracranial pressure. But this method is expensive, very complicated, invasive, and is prone to infection. Hence, there has been emphasis to find new non-invasive ways to monitor TBI, which can detect dynamic change in the brain health, which is portable, easy to use, and can provide continuous monitoring to your brain condition. So we came up with a solution in a kit. Our bioimpedance kit consists of an electrode goggle and an analyzer with a display screen. How it works is the goggles are placed on the closed eyelids of the patient and an electrical signal is passed through the ocular region. Then the impedances are detected over the conduction pathway. And any change in the values that are seen on the graph is the accurate measurement of the brain health. This technique will measure intracranial ICP and cerebral blood flow. And this will also provide us help in decision-making towards the treatment related to TBIs. It will also reduce unnecessary CT scan, prolonged long stays in the hospital, but we will also not miss any changes in the brain conditions.
Currently, there are several ways to assess TBIs. That is blood test, where they look for biomarkers in the brain and can predict if there's a brain injury. But people have fear of needles. Also, you need trained technicians on the site to collect the blood samples from the patient, and then generating the report may take some time. So, this is not very practical. The other non invasive technique is measuring the optic nerve sheath diameter, which is a direct measure to ICP. But this technique also requires sonography, which cannot be bought on the site of incident. And then our biggest competitors, we believe, is BrainScope. Their product measures the brain activity to detect concussions in patients. One of their limitations is that they do not provide comprehensive measurement of the brain, such as ICP or cerebral blood flow, and they're expensive. So as you can see, none of our competitors are ticking all the boxes. But our product AccuSight stands out as it is the most ideal non-invasive. It provides continuous monitoring, it's portable, it provides multiple measurement of brain health with reproducibility and accuracy. And the very important thing, it can be easily implemented in EMT. That's why we believe our product in AccuSight is the most ideal non-invasive brain TBI monitoring device.
All right. So basic research concluded in 2017 for this patent, and our patent was finally approved last month. We plan to license this from the University of Michigan. Our first 3 years, we estimate our costs coming in at about $4.2 million, but we do anticipate grant funding, so we are seeking $3 million from our investors. Out of that $3 million, our first year, we expect a cost of about $1 million. That's going to be for startup, operations, product development, and the following 2 years will be ringing in at $1.6 million each, with $1 million of that $1.6 million figure being dedicated to our phase 1 and phase 2 clinical trials. We do plan to be acquired in 2026 for about $91.8 million, at which point we will break even and our investors can expect a 12 times return on their investment. We can be acquired by any number of these companies listed or not listed. And we would also like to add that our acquiring company has the opportunity to expand their market into sports medicine, combat medicine, and anywhere they see fit, as well as generate a subscription-based service similar to our competitor BrainScope. And of course, we would not have been able to bring this to you without our amazing team. So we would like to say thank you, Cynthia and Fraser. Thank you guys so much. And we are so excited to answer any questions that you may have.
How much do you expect the product to cost?
Cost to make or cost to sell?
Oh,
Oh, so cost to make, it shouldn't cost us any more than about $500 to make. And then based on, we would like this to be anywhere we could see an AED or anywhere there should be an AED. We anticipate selling for a price tag of about $3,000 just to make it accessible. Our competitor BrainScope is selling their product for $25,000.
what do you need? What type of specialty, or do you need to be a doctor to interpret the information? What type of information is coming out, and what level of expertise do you need to be an interpreter?
Okay, sure. So the way we plan to have our device set up is, excuse my rudimentary analogy, but it'll look kind of like an OBD2 reader for our car. You know, you'll have a handheld device with a digital display screen, and we expect to be able to toggle between a screen where you can view like a blood pressure monitor, a pulse monitor. You can have numerical values, and then of course you can flip into a real-time graphical analysis. We would anticipate needing some level of expertise medically, like first responders would presumably have in order to read this, but we do anticipate having within our diagnostic framework an alert, I guess, for thresholds that would be undesirable.
Would you do baseline testing? And is there a way to capture the data so that you could, say, test all the athletes in a school for baseline testing before the season? And what would that do?
Yes, that's exactly how we plan to do it for sports medicine and combat medicine. So we would anticipate that if an institution had access to our products, they would be doing routine testing on, you know, especially for like high school football or college football. Some things to the noggin during practice. So we would expect that continuous, weekly, every other week, you would get baseline measurements for your athletes. And then of course you would be able to monitor if they, you know, did sustain a head injury, that you would see changes in, yes, over time to their measurements.
And is there, sorry to ask so much questions, but is there is it sensitive enough that it can detect subconcussive impacts? You know, what a lineman goes through the course of the season, especially 2000 subconcussive and Pat's gonna see them.
Yes, so we didn't necessarily list all the parameters in our presentation here of what this would be able to measure for you, but this is going to measure cerebral blood flow, arterial blood pressure within the brain. It's going to be able to measure intracranial pressure. So it should, in theory, be able to detect subconcussive injuries in our potential patients. Yes.
So you're using this impedance measurement from across the eyes and through whole skull. I guess I know that's been issued. I guess I can understand if it's a change in intracranial pressure. You'd see an impedance difference at certain frequencies. Trying to understand better about the blood flow. Since blood flow is regional, there could be increases and decreases in different areas, and you have a TBI, you have localized damage perhaps to blood flow. I'm not convinced that I would necessarily believe without seeing the data, which really you don't have, but maybe tell me a little more how it can accurately pick up, small differential spatial changes in blood flow by a simple impedance across the whole head measurement.
So in the animal studies, they were able to push and pull some of this cerebral blood flow and ICP, and some of those measures are actually related. And so I think that was part of what they were trying to show, but they could show that this measure, these impedances and the patterns of the impedances could actually show the differences between some of those different measures. And so I don't know if that answers all of your question, but there were some studies that were showing that you could differentiate those things.
So Ward et al. In 2017, in the paper they have published, where they used animal model, and they saw that the measures that are found by bioimpedances were directly correlated to the ICP measures and also the cerebral blood flow with precision. So yeah, it's based on.
Very much, guys. All right. So next up we have Cal South.
Good afternoon. My name is Blanca. These are my colleagues Noah and Sushmita, and we are the founders of Better CalSal, a solution for PEA. Our company aims to create products that will support the heart to prevent the onset of PEA, and the product that we'll be talking about today is a novel application of a half saline solution, which has half the concentration of sodium chloride in it compared to normal saline. This is very key because the altered concentrations of salt will replicate concentrations found in healthy patients. And by doing this, we can better support the heart and prevent onset of PEA. So to take a step back, what is pulseless electrical activity, or PEA? Does anybody know what PEA is, or has anyone heard of it? So PEA is a condition in which the heart has electrical activity, but it cannot contract. After a sudden cardiac arrest, patients who develop PEA will have a weak or absent pulse, resulting in not enough oxygen reaching the tissues, which will then lead to tissue death within 5 minutes. This then leads to irreversible organ damage and eventual death of the patient. Now, has anyone seen one of these, or does anyone know what this is? This is an AED. So in a hospital, this can be found. For anyone who's seen a medical show such as Grey's Anatomy or House, you might recall a patient who has a heart attack being shocked by one of these and then regaining a pulse and then surviving. From TV, we're given the impression that 90% of patients treated with an AED will survive. However, in reality, this number is closer to 15%. For treatment of PEA, an AED is 0% effective. Heart disease is the #1 killer in America, and the CDC estimates that about 24% of deaths are from heart disease. Of these, 50% are cases of sudden cardiac arrest, and of these, 20% are PEA. 90% of patients who have PEA will die, and 90% will not make it to the hospital alive, and 97% of patients will not exit the hospital alive. Additionally, 10% of all in-hospital deaths are attributed to PEA. To put these statistics into context, that's 356,000 people who will die out of hospital from a cardiac arrest in the United States each year. That's 71,200 estimated PEA events annually in the United States. and that's 64,080 patients who will die from PEA in the hospital. There is a need for a better solution. Ideally, after a sudden cardiac arrest, the underlying cause is found and reversed. But even in a hospital setting, this is tricky because the patient's unconscious and therefore difficult to assess. As we mentioned earlier, tissue death will occur within 5 minutes. The time is of the essence. When EMS arrives, patients are given saline to hydrate the patient and increase the circulating fluids to oxygenate the tissues. However, normal saline has an inappropriate composition of salt, which has been shown to lead to electrolyte disturbances that can interfere with the restoration of a heartbeat. Our solution, Better Cal-Sal, has a more appropriate salt concentration that's representative of the concentration found in healthy patients. It is also affordable as half saline bags already being manufactured at a relatively inexpensive price. Our product is also straightforward to implement because it's just as quick to administer as normal saline, which is what's currently being used, and no additional training will be required.
Moving forward to talk about our competitors in the market. For all cardiac events, CPR and automated external defibrillators are used as a standard of care. But for our pulseless electrical activity, we don't have any standard of care currently available in the market. So a salt water solution like normal saline is provided to patients to increase the contraction rate of the heart, but is not effective and it cannot prevent the sudden cardiac events. But it is used anyway because we don't have any standard of care available for patients. And it's also easy to implement and is affordable. That's why they use it. But we need a better solution to detect those, to improve the quality of life. So we are launching the Better Cal-Sal solution that can effectively increase the heart rate to people experiencing PEA. It can also prevent the contraction rate of heart, and it can prevent the sudden cardiac event. It's easy to implement and is affordable. Now let's talk about our market plan. Our product was patented in 2021, and within a year, we started our phase 2 and phase 3 clinical trial. And this is sponsored by Virginia Tech and Caroline Clinic. We are planning to apply for the FDA approval by 2024. And for this, we will need around $2 million of investment. We are planning to get $1 million of investment from those investors, and we are still seeking for $1 million of investment. We will use this money for some initial approval to upgrade our manufacture and the distributions to enter into the US market by 2027. We are planning to enter into the global market by 2028.
So as Sushmita said, we've already started our phase 2 and phase 3 clinical trials. These are happening in the Roanoke area where we're giving out our product into EMS and into ambulances where they respond to cardiac events, and they're giving either our product or the standard of care, which is just normal saline. We don't have any formal data yet, and that'll happen. We'll get that data back in February of 2023. But the preliminary data that's trickling in is showing that this product is effective at allowing patients to make it to the hospital alive and increasing their chance of survival. Once we get this formal data in 2023, we believe that we can get FDA approval relatively quickly because, as we've said, there's no effective standard of care. And so any tick in the needle that we can offer some help to these patients will probably be accepted quickly. On top of that, we think that hospitals and EMS services will pick this product up relatively quickly because, again, there's no effective standard of care. So we plan to sell our product for $6 a unit, and we think that we can produce this for just $2 a unit, giving us a profit margin of about 67% per bag. We think that we should be able to we will be breaking even in about 5 years. And after that, we will start reaching a larger US market and then a global market. So at this current time, if you have a PEA while you're at home, it's almost certainly a death sentence. We need a better solution, and we have one that's both affordable and is also effective. This is our management team. And then we want to say thank you to to Mason and Kevin Bloomfield, and then of course to Dr. Polzing as well for inventing this fantastic product.
PEA diagnosed, and they always know that 20% has PEA.
So PEA is usually diagnosed in a hospital, and so for the use of our product early on, we aren't going to try to diagnose it as PEA. Pretty much anyone who has a suspected a heart attack, we plan on administering this bag tube because it's safe. So it's not going to hurt any patients who are having a non-PEA, but it can help these patients who have a PEA, which is and it can help those patients.
You give it to everybody.
Yes, everyone who has a suspected cardiac event.
So to that last point in the field, in terms of safety, what if a patient's in diuresis, somebody's hypernatremic, somebody has a congenital heart disease, hypertrophic cardiomyopathy, where they have, abnormal patterns of electrical activity. I mean, a lot of variability. Are you comfortable that in humans, the safety study has ascertained that you would do no damage to any of those patients, or will you collect those types of data in the phase 2 trial?
We will collect those data, and we do expect that there will be some contraindications, such as some of the ones that you just said. But we think that for the vast majority of patients, that the benefit of this would drastically outweigh the risk. But yes, but the phase 2 trials and phase 3 trials that are undergoing will have to evaluate that further.
But in the field, wouldn't it be almost impossible to know whether a patient does have some of those underlying conditions that would lead to contraindication?
For some of them, yes. But we do, and so this is why we will have to undergo these safety trials. But we do think that that the benefit of this will outweigh that. But yes.
And then how much? And I'll use the wrong nomenclature probably, but your formulation. How much leeway does your patent provide? Meaning, could I just make my own Better Call Saul version 2 that has slightly more salt or slightly less salt, get around your patent, but be just as effective?
Our patent does have a wide range of both the sodium chloride and also the calcium chloride, which is the calcium component. So we do have a wide margin within our patent.
Basically, Gatorade and an IV.
Yep.
How many patients are you going to need to enroll in all your trials to get approved?
500 is roughly 500 is our target goal.
And you said you only need $2 million, or you already have enough money to complete the trial?
We have enough money to complete the ongoing trial, but we need the additional funding to ramp up manufacturing and have sales teams and go through the final FDA approvals.
All right. You could
I'm just gonna ask a very quick one. So you're saying that your company's running what's called SPEAR clinical trial? SPEAR.
That is the name of the clinical trial that I was talking about. This phase 2 and phase 3 trial that's ongoing currently. That is the SPEAR trial.
All right, we have one more presentation from Viewnostics.
Good afternoon, everybody, and thank you for being here today. My name is Andrew, and along with my colleagues, Kavya and Katie, we represent Viewnostics, and we're here to present to you a technology to revolutionize the diagnosis and management of intraocular tumors, including retinoblastoma. This is Jimmy. Jimmy is a 1-year-old boy whose life of him and his family is about to change forever. As you can see here, there's a strange finding in his right eye. It's an opacity called leukocoria. This was identified in a pediatrician's office. He will subsequently be sent to a pediatric ophthalmologist and diagnosed with retinoblastoma. So retinoblastoma is a rare form of eye cancer with a strong genetic component. It represents 2% of all childhood cancers. Currently, there's around 300 cases per year in the United States, with several thousand that occur annually worldwide. The condition is rare. However, the indirect economic costs from impaired and lost vision over time are substantial. As you can see on the map, these are a list of all the dedicated retinoblastoma treatment centers across the world. You'll notice that there's a paucity of them worldwide, particularly in areas of high incidence, such as the United States and India. This is a gap that our technology will be able to fill effectively. Current diagnostic technologies, including MRI, ultrasound, and electroretinography, are limited in location and accessibility. Biopsy for the tumor in order to identify these prognostic markers is contraindicated because biopsying a retinoblastoma can cause metastasis. Finally, current treatments require usually removal of the eye, hence an onset of vision impairment and vision loss. And so we here at Biomedical want to develop the next generation of retinoblastoma prognosis and treatment through minimally invasive tumor characterization.
Thank you, Andrew. So I'm going to be talking about our solution to this very pertinent problem. We have licensed a highly researched and published technique through labs at USC that allows to diagnose and stage retinoblastoma through biomarkers found in the aqueous humor. So our company will bring to market an assay kit to use this aqueous humor biomarkers for clinics across the United States. This assay is going to be similar to that we've seen in COVID tests and pregnancy tests. It's called a lateral flow immunoassay, where we will be providing the actual test strip as well as the solution needed to be mixed with the aqueous humor to place on the sample pad and then garner a positive or negative result. Our company is the sole owner of the rights to use the aqueous humor in these assays, and this allows for a minimally invasive, especially compared to the enucleation of an eye technique to diagnose and stage retinoblastoma, with the presence of these biomarkers predicting eye salvage significantly better than clinical staging, as shown in our Nature review paper. As far as our competition goes, our main competition is the current standard of care, which, as Andrew mentioned previously, is comprised of MRIs, ultrasound, and electroretinography. While this is minimally invasive, its prognostic power for eye salvage only relies on seeing if the tumor grows over time, in which they will enucleate the eye and in some cases provide intensive chemotherapy. This is not an accessible or affordable option for most patients. We are aware of an additional Chinese IP that utilizes nanoprobes and nanoparticles as a method for diagnosis, but there's very few data that is supportive that we were able to find for this particular patent, and we do not think that it will stack up to our company. So going back to this map, our existing market is very small within the United States, and so we plan on expanding this market through the use of HRSA to over 14,000 health center grantees and service delivery sites. This will allow for our product to be able to be more accessible to patients like Jimmy, as well as his siblings, and to the additional eyes that may not look like they are impeded at the time, because the earlier a diagnosis is able to be acquired, the easier it is to be able to salvage the eyes and the vision of these children.
Alrighty, thank you, Katie. So I will be going very briefly into the developmental milestones for Biomedical. So we are envisioning completely licensing our IP along with starting our research lab and applying for federally funded grants in 2023. During this process of our research lab, we aim to have our assay kit completely developed during the first 6 months of 2025. And we will immediately start phase 1 clinical trials before we start our national distribution in 2027. So in our Biomedical, this is what we are asking you as the investors. Our first 4 years will be actually starting our research lab from the ground. We are envisioning about a $9 million use across those 4 years, and we will be planning on subsetting this between both grants and investments, currently at a 50-50 ratio. These are some of the federal grants that we will be reaching out to. The FDA and the NIH have calls for action currently for increasing research and therapeutic opportunities for patients that suffer from rare diseases, including retinoblastoma. We will also be contacting the HRSA organization to help us with distribution of this product when it gets to that point in time. So we envision that our sale price for each of these assay kits will be about $3,500 a piece, but our cost of goods will be about $100. Given that retinoblastoma is a rare disease, we're envisioning currently selling about 2 units per clinic and reaching about 10% of the market in 2027. But this will still yield a cash positive result, and then expanding out to 2029, about 40% of the US clinics to yield about $60 million of a cash positive before expanding to international sales. Like Andrew had mentioned, there is an issue in India in 2030. And so we do acknowledge that we do have acquisition potential down the line. These are some examples of companies that also do work in the liquid biopsy and cell-free DNA space. And we do acknowledge that potentially down the line, we could be acquired by them. And great science cannot be just done by us three alone. We are incredibly grateful to have the support of various different clinicians around the US that have been giving us valuable advice so far. Special thanks to Dr. Jessie Berry, who is actually our inventor and is willing to stick around with us as we develop our product and help us implement this in various different clinics that she's oversees as well, along with Dr. Fasciani, who is a pediatric ophthalmologist here in the Roanoke area, who was able to give us prevalence information about retinoblastoma here in the area. And lastly, thank you so much again. We really appreciate your time, and we are Viewnostics advocating for better eye health for all. And we are open for questions. Yeah, Dr. Friedlander.
For that, so that's fantastic. But my concern is, you pointed out that's, I believe, 300 cases per year in the US. You also said you were going to have 14,000 US sites. So I calculate that's a 42-to-1 ratio of sites per cases. Then you said you're going to give 2 kits, I believe, each site. So that's 28,000 kits for 300 cases. I think where you can see where I'm going with this. There's going to be a challenge economically in this. And so what I'm wondering on the positive side is, could you have thought about expanding this assay of the aqueous for other things as well in terms of eye disease? Because I think if you're limited to this market, you're running right into those numbers, right?
That's a fantastic question. So there's already some early indications that we can expand into uveal melanoma, as well as vitreoretinal lymphoma. So particularly with uveal melanoma, when we discuss it with ocular oncologists, there's a particularly big need to be able to subtype these tumors, whether it was a primary intraocular tumor or a metastasis from somewhere else in the body. And being able to subtype, you can both identify prognostic markers, you can identify its particular molecular characteristics, and then tailor precision therapies. And so that will be sort of the second phase as we begin to ramp up our assays and distribute them for retinoblastoma. The research lab in part will take on roles in developing the technology for uveal melanoma specifically.
That's great. So just to follow up for a second, the number of cases is one thing, 300 for retinoblastoma plus these other things, but the number of suspected cases that need tested is going to be greater. So that's my question. How many tests are needed well above the 300 confirmed cases based on suspected anatomy and pathology?
I did some math on that for this presentation, and I've determined that through testing through both eyes, because sometimes it only shows up in one eye, so it would probably be in best interest to test both, as well as testing siblings in cases that are not retinoblastoma, that we would need about 5,000 tests per year for these 300 cases.
What's aqueous humor? What's aqueous humor?
The aqueous humor is the liquid that keeps your eye round. It's pretty much the liquid that fills up the inside of the eye. And
okay, so basically, all these kids get sort of light scope tests. When something looks wrong, they then how do you get that?
You have to take a biopsy through a needle in the eye, but the alternative to that is to completely remove it.
Okay, so basically, help me understand, what does it tell you when you hit the liquid, you run it through the biomarkers, and then what do you do with the resulting information?
So the resulting information would allow for an earlier diagnosis, as well as the ability to figure out how bad the particular case is. So a lot of the times with staging, enucleation is not needed. And if any signs show that the retinoblastoma may be getting worse, doctors default to the enucleation, even though it's not always necessary. And that's the main issue that we're trying to address.
Thanks.
All right, that's going to have to be the last question. All right, so I want to say thank you to all of our teams. You did a really great job. Like Dr. Gordy said at the beginning, it seems like every year the presentations get better and better. We are a little ahead of schedule, so I'm going to have to find something to talk about for the next 15 minutes. So no, all right. So the judges will follow Linda to where they will convene to determine our winners. Everybody else, there is a reception out in the atrium. And Dr. Gordy, do you have anything to add?
Yes, there was one very important person I forgot to thank. And that, of course, is my wonderful administrative assistant, Linda Collins, who. So thank you, Linda. All right, as Zach said, the judges will retire to the room that Linda has prepared for them, and there's a reception outside. So well done, everyone.